Okay, in this short tutorial, I will be discussing the source sink relationship, or what you call as the food transport, the translocation of the food via phloem bundles. Okay, so we know the source we call that part of the plant where the food is synthesized, and we substantially know that the leaves are the photosynthetic units in almost all of the plants. Yeah, there are some plants which can let the other parts to become the photosynthetic units, but generally speaking, the leaves act as a photosynthetic unit. So the leaves will be performing the photosynthesis and will be preparing the food for the plant. Let's suppose I will be here showing the food in the form of little short circles. Let's suppose this is the food prepared by the leaf through the process of photosynthesis, and this leaf acts as a source because it's here that the food is synthesized. Any part of the plant where you are synthesizing the food, you will call that as the source. And it depends upon then the plant you are discussing about, because there are some plants which store the food inside the roots, some store the food inside the stem, and some transport the food to the fruits for the ripening purposes. Okay, now let's suppose here I'm talking of the plant where I need to transport this food from source to the sink. Sink means the part where you are actually transporting the food, the part where you are storing the food. Here, for example, this is my own choice. I'm taking the food from the source to the sink, and sink I am labeling here as root. So root is my sink. Okay, so my purpose is to transport the food from the source leaf to the root, that sink. So this is the source sink relationship. How come we can go about transporting the food from the source to the root through these distributing phloem bundles? Okay, well, the leaves are preparing the food. Once the food is stored inside the leaf, it needs to get passed on to the neighboring phloem bundle. Phloem bundles are always scattered throughout the plant tissue, plant whole plant organization. Inside the leaf there are also some phloem bundles. Let's suppose this phloem bundle may also be present in the leaf, but just for the understanding, I'm just showing it here, I'm just representing it here, a part distance from the leaf. The food is stored inside the leaf. Now this food will get transported to the nearby phloem bundle. Okay, so this food is now transported near the phloem bundle, which is which is at a short distance from the leaf. So this transportation of the food from the leaf to the nearby phloem bundle requires energy. That means you will be utilizing an ATP molecule. So when you utilize an ATP molecule, you will be dissociating the ATP into ADP plus inorganic phosphate. When you are just hydrolyzing the ATP into ADP and inorganic phosphate, some energy is released. That energy is utilized here to transport the food from the leaf to the nearby phloem bundle. Now this food, actually our ultimate goal is to transport the food from the from this phloem to the ultimate root. Okay, but for that purpose we need to go through these bundles as well. How come we can go about doing that? So there should be some source here in this phloem where we have transported the food, so that that source could push this food to this phloem, then to this phloem, then to this phloem, then to this, and then ultimately to the root. Since here inside this phloem we are having the food particles, that means solute particles. So the solute particles are very high. That means the concentration of the solute is very high, and we know there is least amount of water present inside the phloem. Yeah, there is a little amount of water, but not that much. If we talk of if we compare that as that of the solutes, the concentration of solute is very high and the concentration of solvent is very low in this phloem bundle. That's why we are creating here an environment known as hypertonic environment. That's where the solute concentration is more and solvent concentration is less. So there is yet no source found which could push this food from this phloem to this phloem. How come we can achieve this source? Well, adjacent to this phloem, that's what these are the phloem bundles. Adjacent to the phloem, you will always find a xylem lying parallel to it. Okay, so this is the xylem here. I have just represented it with a whole big massive circle. Let's suppose we know the xylem contains bulk amount of water. So this is water present inside the xylem, and this is very basics of the biology. that xylem is responsible for the conduction of water. So the water is present inside the xylem. Inside the xylem, the concentration of the solvent is more because water is more, and the concentration of solute is very less. This is in contrast to that of this phloem bundle. Here the concentration of solute is more, concentration of solute is less, solvent is less, hypertonic environment. Concentration of solvent in the xylem is more, concentration of solute is less, so hypotonic environment. And remember, the property of the water is to move. Water will always prefer to move from hypotonic environment to the hypertonic environment. It's pretty basics of the biology. You should know this, that the water loves to move from the hypotonic environment to the hypertonic environment. So the water will flow like this from xylem to the phloem. Okay, now the water has entered this phloem bundle where the food is already present. So now the water molecules are also present inside the phloem bundle. With the presence of the water, the phloem bundle is creating a hydrostatic pressure, because the water has moved from this xylem to the phloem through the process of osmosis. So the water present inside this phloem creates a hydrostatic pressure, and this hydrostatic pressure helps the food to pass on to the nearby phloem bundle. Now you might be surprised, why this happens, because in this phloem we don't have any food, we don't have any solvent molecules or solute molecules. This is just empty, and the pressure here in this phloem is very, very high. Hydrostatic pressure is very high here in this case. In this phloem, the hydrostatic pressure is very least. So there is pressure gradient. This pressure gradient helps this food to pass from this phloem to this phloem. Now the food is present inside this phloem along with the water. Now hydrostatic pressure is developed here in this phloem. Compared to this phloem, this has very high hydrostatic pressure. This has very least hydrostatic pressure. Once again, pressure gradient is created between the two phloem. This pressure gradient will push the food from this phloem to this phloem. Now the food is present inside this phloem and along with the water molecules. Now hydrostatic pressure being developed in this phloem. So comparing this phloem with this phloem, this is high hydrostatic pressure, this is least hydrostatic pressure. Once again, the pressure gradient formed, it pushes the food from this to this. Same case happens from this phloem to this phloem. At the end, we are having solute molecules, food along with the water in this phloem. Now the food which needs to get to the root, to the sink, will get transported from this phloem to the root. It also will require the energy, hence is an active process. It's an active process because the food from this phloem will be transported to the root by consuming some ATP molecules, hence is an active process. But the water is left behind. Remember, we don't need water get stored inside the root. We just need only the food to get stored in the root. So the food solute particles are transported from the phloem to the nearby root, and the water is left behind. And this water will then move back to its original source. It will move back to the xylem. And we have recycled the water back to the xylem. The water is present in the xylem, and the food has been transported from the source to the sink. This is how the source-sink relationship works. I hope it helps you understanding this topic. Thank you.